Hi, this is Dr. Jason Lee, clinical immunologist and allergist practicing in Toronto, Ontario.
Hello. In this lecture, we're going to be talking about immunoglobulins or antibodies. As previously discussed, antibodies are produced by B cells. They consist of two light chains and two heavy chains. These are denoted as H and L. Below the hinge region of the Y are the CH2 and CH3 regions, which determine the Fc portion. On the tops of the Y are three regions called the complementary determining regions. These are all within the top portions of the Y called FAB segments. The complementary determining regions really determine the affinity of that antibody. Now, immunoglobulins have two roles. One, to act as an antigen receptor, and two, to activate effector functions depending on what is needed. The antigen receptor function selectively recognizes and binds antigens, such as toxins, viruses, and molecules exposed on the surface of pathogenic organisms. This is a variable domain function. In the effector function, this is largely correlating to the C domain function in CH2-3. This will either activate complement, inactivate the foreign antigen, or cause lysis of cells, or cause opsonization. The human immunoglobulin kappa locus and lambda locus determine various affinities. They undergo genetic rearrangement by a process known as inversion or deletion to randomly mix up the various segments and gene sequences to come up with random combinations of affinities. Like any other gene that eventually gets made into a protein, this undergoes transcription, mRNA production, initial polypeptide production, and maturation of the chains. The kappa and lambda locus contain a V variable and a J joining gene segment. The human IgH locus contains a diversity as well as V and J gene segments. This contains multiple different C genes to determine the different effector functions of that antibody. Now, the primary structure of the heavy chain variable domain consists of three CDR regions that are formed by VDJ rearrangement. The CDR3 region, I'll emphasize, is key for somatic hypermutation, which is involved in affinity maturation to produce better antibodies so that the second time you see an infection, you can mount a higher, better quality response. Further diversity is added to antibodies with a process called non-homologous end joining. This is accomplished by one of four ways. One is site-specific cleavage by RAG1, RAG2, HMG1 and 2, recognition of cut ends by DNA PK, Ku70 and 80, repair of the ends with XRCC4, DNA ligase 4, and Artemis, and modification of repair with TDT. All of these things will add further diversity. A deficiency in one of these processes can lead to severe combined immune deficiency. There will be more on this with Dr. Kim's lecture coming up. There is occasionally combinatorial diversification and junctional diversification terms that you'll come across. Again, these are different processes used to exponentially increase the diversity of antibodies that your body can produce. Now, a few definitions to go by. A paratope is the part of the antibody that binds antigen. An epitope is the part of the antigen that is bound by the antibody. Because antibodies can bind different epitopes on the same antigen, antibodies can recognize fine differences between antigen, i.e. discrimination between pig and human insulin. Because of similar epitopes can be found in different antigens, some antibodies bind more than one antigen. This is termed cross-reactivity. Molecular mimicry occurs when an antibody recognizes something that is similar to the thing that it wants to attack. There's been a lot of recent work on Guillain-Barré, a neurologic disorder, as an end result of molecular mimicry from certain GI infections. Let's talk about class switching, a major concept that I want to address. During an immune response, the B cells start off with the IgM antibody, which acts as a receptor. Let's say an antibody needs to deal with an extracellular infection. Well, ideally, you would want the antibody to activate parts of the immune system that deal with this kind of infection. This is where class switching is important. B cells can produce IgG, A, E, or D with the same antigen specificity as the original IgM. In a subsequent infection, though, the antibodies that are selected to survive will be of even higher affinity as they undergo somatic hypermutation. Effector functions, as I discussed, involve complement fixation, complement activation, and binding of the Fc receptors, which are found on various WBCs, including mast cells, basophils, monocytes, platelets. Eosinophils also contain Fc receptors too, which is important in asthma. If we look at the biological properties of these different subclasses and classes of antibodies, IgG can be broken down into 4 subclasses. The bulk of all antibodies in the body is IgG subclass 1. This is largely produced to induce complement fixation. This is also used to protect the fetus in fetal development and crosses the placenta. It also binds to white blood cell lymphocytes, macrophages, neutrophils, and platelets. The half-life of these is typically 23 days. Now, in decreasing order, IgG subclass 2, 3, and 4 are also produced in bulk. All of these can activate complement fixation except for IgG subclass 4. Alternative pathway of complement fixation is accomplished by 3 and 4. Binding to lymphocytes occurs with all 4 of these subclasses. IgA is also broken down into 2 subclasses, 1 and 2. The vast majority of IgA is subclass 1. Both IgA subclasses largely work with neutrophils and complement fixation. IgM is also good at complement fixation and binds to lymphocytes. IgE is largely thought to fight parasitic infections, and it fixes onto mast cells, basophils, and eosinophils. The half-life of IgE is roughly 2.5 days. Effective functions of human isotypes are 2 ways that antibodies can induce killing. One is by complement lysis, and the second is by antibody-dependent cell-mediated cytotoxicity, which is directly Fc receptor driven. Last concept to discuss is glycosylation. All antibodies are glycoproteins. The sugars, or glycans, bound to the Fc domain can vary between individuals and vary within the individual. There are as many as 32 plus different patterns. The presence or absence of these sugars affect the structure, stability, and function of the antibody. Binding to Fc receptors can be affected, and as such, antibody-dependent cellular toxicity is determined by glycosylation in some instances. Binding to complement can also similarly be affected as well. The function overall is affected by glycosylation. The specific patterns of glycosylation is beyond the scope of this lecture. Thank you.